We have an epidemic on our hands. And I'm not talking about COVID-19, but instead about the chronic pain epidemic that has been sweeping across North America for decades. About 1 in 5 Canadians suffer from chronic pain. Pain destroys relationships, careers, and lives. Treating chronic pain is extremely difficult. Treatments are often ineffective and can have dangerous side effects, including addiction. According to the Government of Canada, 1,018 people died from opioid-related deaths between January and March of this year. That's 1,018 people in 3 months. But I may not even have to tell you this. In all likelihood, you or someone you know is suffering from chronic pain, and you may have seen the way that this disease destroys families and takes lives. There's good news though. We have amazing teams of researchers worldwide that are working tirelessly to learn more about pain and to develop better treatments. But we have one more problem. Our fundamental knowledge about pain physiology is based almost entirely on studies in male rodents. Scientists need models to study pain. We can't just test on or collect tissue from regular people, but models are inherently flawed and imperfect. What we do need, though, is sex-inclusive basic science research and for ways to test new therapeutics on human tissue before moving to clinical trials. And this is because over 95% of chronic pain clinical trials fail. And I think that's because our preclinical research models need improvement. What my thesis has done is help bridge the gap between basic science rodent models and clinical trials. Thanks to my 2 amazing supervisors, Dr. Mike Hildebrand from Carleton and Dr. Eve Tsai from the Ottawa Hospital, we've developed techniques to look at human tissue from organ donors. When the family of an organ donor consents to donating their loved one's tissue to our study, we collect the spinal cord, which is a super important part of the pain pathway, directly from the operating room. The patient is deceased, but the cells inside the spinal cord are still firing, communicating, and working together and with each other. We can then take this tissue, run tests, and compare them directly to our rat models. This preclinical human tissue work has yielded incredible findings. Because we get male and female organ donors and work with male and female rats, we can compare between sex and between species. What we've found is that the targets that we've looked at are conserved between species, but differ between males and females. These differences have huge implications for how we treat pain. We're so lucky to get to do this research, and I'm really excited about our preclinical human models of pain. I hope that this work leads to better treatments for the millions of people worldwide suffering from chronic pain.